Okay. I'm gonna resume my presentation. Okay. Again, let's try to concentrate to the important things of the day. We are going to be talking about Thrips parvispinus and provided you with more research updates. As we mentioned, the worldwide distribution remains very similar to the previous months, with the pest being present in Southeast Asia, Oceania, Europe, some parts of Africa, as you can see, and north part of the Americas and the Caribbean. In the United States, the pest has been found in Hawaii, Pennsylvania, South Carolina, Ohio, North Carolina, Georgia, Florida, and Colorado. And the detection in Florida was in 2020. As a few days ago, the Thrips parvispinus obtained an official common name from the Entomological Society of America. It is now called short spine thrips, and this is the name that we are going to be using moving forward. What you need to remember is right now, the short spine thrips is not a regulated pest, and nurseries that have large active thrips populations are going to be the ones that are going to be quarantined. If you take a good action maintaining the thrips populations to a bare minimum, or even if you can actually eliminate the thrips population from your property, you will not have issues. If you have more questions about the regulation of this pest, feel free to reach to Mrs. Mary Reed and Mr. Charles Gonzalez. I have inserted the contact information here. So let's start with the biology. The short spine thrips, as you all know, it's a very small insect, and here you can see the pictures of the male and the female. The life cycle of this insect pest is fairly simple. So we have the adults that they mate, and then the adult female lays her eggs inside the plant tissue, and this is why we cannot actually detect the eggs. When the eggs hatch, we have the emergence of the first instar larva that molts to a second instar larva. And the second instar larva moves towards the end of this stage. It moves to the soil to pupate. So as soon as the second instar larva moves to the soil, it molts to a prepupa, then to a pupa, and exits the soil as an adult. We did a series of experiments that were led by Isamar Reyes, who is a master student in my lab. And Isamar actually explored the effect of temperature on the short spine thrips development. What we can see in this graph is the average number of days that the thrips require to complete their immature development, so from the egg stage until they become adults. And you can see that the lower the temperature, the longer it takes them to reach the adult states. But if the temperature is high, like 90 Fahrenheit, then the development goes very fast. You will see that the temperature of 72 Fahrenheit and 80 Fahrenheit was in the middle. But again, these were also favorable temperatures for the immature development. Then we had a look at how long the life cycle takes to complete. So when the females lay the first egg, until this egg reaches adulthood, and that female is about to lay her first egg. So until this circle is being complete. And we found out that at 90 Fahrenheit, this life cycle takes approximately 10 days. But at 80 Fahrenheit, the life cycle takes 12.8 days. If the temperature is a little bit cooler, so at 72 Fahrenheit, the life cycle becomes a little bit longer, and it is 18.4 days. And when the temperature is much colder, so at 63 degrees Fahrenheit, then the life cycle takes 37.4 days. So we can see that temperature really has a significant effect on the life cycle of the pest. And something that I didn't mention, all this information that we are delivering today regarding the biology was performed in constant temperature. So it was one temperature throughout in the day and night. So there was no fluctuation. So these are lab ideal conditions. When we looked into short spine thrips longevity, so how long the adult thrips lived, we also saw a significant effect of the temperature. So we can see that the lower the temperature, so at 63, the longer the adult males and females live, especially if we compare to 90 degrees Fahrenheit. Another interesting aspect of these experiments that was revealed is that the females live longer than the males. So you can see that all the pink bars are higher than the blue ones that correspond to the males. Of course, a burning question in the beginning was how does short spine thrips reproduce? And we saw that when the females mate with the males, then they can produce offspring, so other insects of both genders. They can produce females and they can produce males into 3-to-1 ratio. So for every 3 females, we had a production of 1 male. But when the females do not mate with a male, do not have access to a mate, then we saw that again they can reproduce, but this time all their offspring are males. And in this case, a sex ratio, it's 100% males. This mode of reproduction is called Arenotochus parthenogenesis, and the insects use this in order to overcome the obstacle of reproduction when the males are not available. So in this case, if females do not have the presence of male, what they're going to do, they're going to produce sons, and then the sons will mate with the mother, and then the mother will start producing sons and daughters. So when we looked into the short spine thrips fecundity, which is the number of eggs a female can lay per day, we saw some very interesting results. Again, we saw that the temperature affected how many eggs each female can lay per day, and we saw that the moderately warm temperatures, so 72 Fahrenheit and 80 Fahrenheit, were actually very favorable for the reproduction of this pest. The females laid significantly more eggs, viable eggs, in comparison to the extreme temperature. So a very low temperature, like 63 degrees Fahrenheit, or very high temperature, which was 90 Fahrenheit. Another thing that we wanted to explore was what happens if the thrips do not have access to their primary food source, which would be plant material. So we keep our colony on beans, and we did an experiment where we had as a control leaf discs of beans that we offered to the thrips to feed, but we also placed the insects in petri dishes where they didn't have access to any food source, or we provided them with pollen, or we gave them water that had sugar at 1% concentration, or just simply water. And we observed how long they were going to survive. And as you can see from this graph, when they had access to their primary host, which was the plant tissue, right, they survived for almost 13 days. However, when they didn't have access to that tissue, and they were given an alternative food source or no food at all, the thrips survived only 1 day. An important question that was very evident since the first interception of these thrips in the nurseries was how much damage the thrips can actually cause. So Isamar started doing some experiments to really assess the damage on different hosts, and she worked on vegetables host as well as ornamental hosts. And I'm going to give you a series of photographs where you're going to see the feeding damage caused by a first instar or second instar larva and the adult thrips. And she also, what Isamar did, she also put different densities of the thrips. So she had leaf discs like this one, where she didn't place any thrips, and this was the control. Then she had a leaf disc where she placed only one individual, or 5 individuals, or 10 individuals, and then she scored the damage. In order not to overwhelm you with a lot of graphs, today I'm just going to show you the pictures to visualize how the damage looked like. So these are the results for the beans. So as you can see, when first instar larvae were fed on beans, of course, if the more larvae we have on a given surface, the more the damage that we're going to observe. Similar results for the second instar larvae. Again, at the higher densities of 5 and 10 individuals, we can see a clear scarring of the leaf tissue, while in the first, only when we have one individual, the damage is smaller. But when we take a closer look to the adults, we can see clear difference that the adults, even when we have only one individual feeding for only 24 hours, we can see a clear damage. Of course, at higher densities of 5 and 10 individuals, thrips are very aggressive and they cause a lot of damage to the tissue. Now I'm going to show you similar set of pictures, but for peppers. Pepper is also a preferred host for these thrip species. So we did the same thing, but using pepper leaves. And as you can see, the first instar larva, again, especially at the density of 5 individuals and 10 individuals, they scarred the tissue a lot more than when we had one individual. And all this data are feeding for only 24 hours. This damage that you see is only caused within 1 day. The second instar larva, it showed a similar trend. Again, the higher the density, the more evident is the damage on the leaf tissue. And again, the adult, even when we introduce a single individual, can cause a very strong damage, a very strong scarring to the leaf tissue. And you can see also, of course, that the higher the density of the adult females on the leaf tissue, the higher also the observed damage. Then we used also gardenia as a host, and we repeated the same. Gardenia has a different type of leaf. The leaf is waxy, is thicker than a vegetable host. And as you can see, when we introduce one individual first instar larva, we couldn't really detect a lot of damage. But as we increase the density from 5 or 10 individuals, we can start seeing scars appearing. Very similar results when we released the second instar larva. In the beginning, with one individual, we didn't see a lot of scarring occurring, but as soon as we increased the density to 5 or 10, then we saw scars appearing on the tissue. The adults, once again, even at only when we introduce one individual, we could see scars appearing in the tissue within the first 24 hours. Then we also explored hibiscus, because hibiscus has been reported as a host, and we did the same experiment. And again, we observed that because of the structure of the leaf and the quality of the leaf, the scarring occurs, but is not as evident. And we need at least 5 individuals in order to start seeing small change of color of the leaf. Same results for the second instar larvae. We really need to have at least 5 individuals feeding on the leaf in order to observe damage within 24 hours. And of course, if we have 10 individuals, the damage is more evident. But in the case of the adult, the damage was quickly observed, even at 5 individuals. And with one individual, again, on hibiscus was a little bit more subtle. But as you can see, again, when we released 10 individuals, we had a lot of scars, and it was very easy to score the damage. We are still collecting data on Mandevilla, and something that I should mention, all the thrips that were used in these experiments, they were adapted to the host. So they were reared at least for one generation on the corresponding host. So they were familiar with the host, so that when they were offered the host, we can really see immediately the damage that they can cause on it. And this is important information. So what we can conclude from the biology is that section is that the short spine thrips pupates in the soil approximately at 1 inch depth. I didn't show you the data today, but this is something that we have said in the previous webinars. I need you to have this in the back of your mind because it affects how we approach the management of this pest. Additionally, what we saw today is that with increasing temperature, the development is faster, and we observe slow development at low temperatures. We also saw that adult females can only survive for 1 day if they do not have access to their host. And we saw that the adult female is the most damaging stage, followed by the first instar larvae and the second instar larvae. Scarring is more evident on vegetable crops that they have a tender leaf tissue in comparison to ornamental crops like gardenia and hibiscus, where the leaves are a little bit harder and they have wax. So with this, I'm gonna move towards the management portion of the talk. We have identified several management tools that we want to assess against the short spine thrips. Today, I'm gonna give you a very short recap of the contact insecticides and the deep treatments for cuttings. Most of the talk is going to be focused on the use of entomopathogenic nematodes and fungi. And then Paola is going to talk about the potential of predatory mites, while Livia is going to talk about the potential of predatory insects. We are not going to address the monitoring aspect at this point because there's still research that's being conducted. So, as a short recap regarding chemical control of the short spine thrips, this information is not new. I'm just going to refresh your memory. We started when the pest was regulated a couple of years ago. We started with some lab evaluations, and we targeted the adult stage, the first instar and the second instar larvae, and we used contact insecticides. We tested 21 conventional insecticides and 11 biorational insecticides that include horticultural oils and insecticidal soap. For the lab assays, we used a leaf disc from a bean plant, and on this leaf disc we introduced 5 individuals from either stage of the thrips, and we evaluated 2 application types. A direct application where we directed the insecticide on the thrips, or a residue toxicity application where we directed the treatment on the plant and then we released the thrips. Due to the fast turnaround at this time, when the nurseries were quarantined and they had the option to be re-inspected within 48 hours, we were trying to find treatments that can have the knockdown effect. So we assessed mortality within 48 hours, but we also assessed the feeding damage caused by the thrips within these 48 hours. Overall, from the lab assays, we identified actually several formulated products that they were available to be used in the Florida market on ornamental plants, either in nursery and or greenhouse conditions. And you can see that these products, they could be efficacious towards either first instar larvae, second instar, and adult. And most of them had very good efficacy when they were in direct contact with the pest or when they were applied on the leaf tissue and then the pest fed on them. You will also notice that there were some products that they were still showing potential because although they might have not necessarily killed the pest, they stopped the pest from feeding. So we could cease observing damage. And we were also able to identify also some promising biorational insecticides, and most of them are horticultural oils, and then peat is a soap. And taking all this information, we did some follow-up greenhouse evaluations so that we can use plants and test efficacy in a more realistic environment rather than a simple leaf disc. So, of course, we could not test all of these different treatments, but what we did, we selected the 9 best products that included conventional and biorational insecticides. We included water as our control. And then we did a greenhouse experiment, but we assessed both types of spray application. So we did a curative spray application where we had active thrips infestation, and then we applied the treatment. But we also did a prophylactic spray application. So we had a clean plant without the thrips. We first applied the treatment, and then after 24 hours, we released the thrips. As I mentioned, water was our control. We had 6 plants per treatment per spray type and plant species, because I mentioned plant species because we worked with mandevillas and gardenia plants in its trial. And we replicated this experiment twice. And we also, in the case of mandevilla, we also had 2 different cultivars that we could incorporate in our essays. So today, due to the limited amount of time, I will show you the thrips mortality data for the mandevilla. So this is a similar graph that I have showed you in the past. If you see that the code is reddish or brown, it means that this treatment was not more effective, more efficacious than the water control. However, if you see that it is marked as blue, it means that the treatment was more effective than the water control. The circles signify the direct spray application, like the prophylactic, the curative, and the asterisk, the prophylactic application. So in this graph, you will see vertically all the different treatments that we had. The water is being used as a way to compare the treatments. This is why it is not displayed. Then we have on the y-axis the mortality in percentage ranging from 0 to 100%. And as you can see, again, we had a lot of variation, but chlorphenapir, it was the most efficacious product, causing up to 80 or even higher in some cases percent mortality. But we also saw that conserve and timectin, which is abamectin, you can also find it as avid, these 2 were also very efficacious towards thrips in both exposure routes. Expire, as well as the mineral oil and the sesame oil, they had significantly higher efficacy compared to the water control when they were used in a prophylactic manner. So when we sprayed the plant and then the thrips were released. While Overture, it was the opposite. It had significantly higher efficacy than the water control when it was sprayed in a curative manner. We didn't stop there, and currently Paola Villamarin, that you're gonna hear from her in the next talk, She also explores the potential of essential oils against the short spine thrips. These are oils that they are not formulated just yet. They are not available in the market, but we want to see if there is something interesting that we can further explore. So what Paola does, she has, in collaboration with USDA ARS, they have identified some essential oils like manuka, copaiba, ginger, pepper tree, tea tree, and these oils are being diluted into absolute ethanol. And she does bioassays in the lab, testing them against thrips, using all these oils. And of course, as a control, she has two controls. She uses the absolute ethanol and a non-treated control. She also tries to identify which is the right concentration of each essential oil in order to achieve high mortality, and she tests for 0.01%, 0.1%, 1%, and 10%. The target states that Paola is working on is the second instar stage. This is the longest larval stage, and we saw from the feeding assays that is a stage that can cause significant damage also to the plant tissue. So she has arenas that they look like this, as you can see in the photo. She places a second instar larva, and she applies the treatment using a spray water tower. And she scores mortality every 24 hours again for 2 days, and also assess the feeding damage. And she replicates this many times. She has 30 replicates every time, and she replicates at least 3 times more. So in total, it's minimum 90 replicates per treatment. So this is like a graph showing the first results that she has obtained. We have on the y-axis the proportion of that second instar larvae over the different essential oils, as well as the non-treated control and the solvent. And what stands out is that, of course, the higher the concentration of the essential oil, then the higher the mortality, with manuka at 10% and ginger at 10% really standing out from all the essential oils. Additionally, when we looked into the thrips damage, the results vary, but what caught our attention is, again, that in the case of ginger and manuka, due to the high mortality that they caused at the 10% concentration, we saw a significantly less damage, less scarring on the leaf tissue. And all these data are within 48 hours. So we continue this research, and hopefully we will be able to refine this information, test more essential oils, and provide you with more updates in the future. So for the chemical control, we have identified at least 12 conventional and 3 biorational insecticides that show potential. The greenhouse evaluations validated the lab results for the most efficacious active ingredients on Mandevilla. This is what I showed you today. We also saw that the mineral and the sesame oil cause significant mortality when compared to the water control if they were applied preventatively. Additionally, we saw that thrips target, we didn't actually see that, but from these experiments, we were also able to see that thrips target primarily the upper part of the canopy. And efficacy may vary with the host plant. And we have already seen these efficacious active ingredients yielding different results if we change from Mandevilla to Gardenia. But today I didn't have time to go through these details. Just as a reminder, how to avoid resistance, implement multiple control methods, okay? Do and implement an integrated pest management program. If you only apply chemical control, you're going to make the problem worse, bigger. Only chemical control is not going to solve the issue in the long term. So you should try to rotate products with active ingredients that belong to different IRAC groups. And when we say IRAC groups, we need to be careful because sometimes some products they might have multiple active ingredients and they can overlap, so we need to be careful how we do these rotations. And for example, if you apply expire, you cannot afterwards apply conserve because they share an active ingredient from the same class. The same goes for Pradia and mainspring. You cannot put them back to back. Another aspect that we have previously showed is the deep treatments that we developed to disinfest cuttings of the short spine thrips. I'm just going to do a recap here. What we did, we tested products that in the label allow for dipping, like Safoil X, which is a mineral oil, and 3 formulations of entomopathogenic fungi, all of them belonging to the species Bavaria bassiana, different strains of Bavaria bassiana. We worked with bean cuttings, and this graph shows you the proportion of dislodged thrips when they were dipped gently for a few seconds into this treatment. We also included water and one treatment where the cuttings were not dipped at all. As you can see, the most efficacious treatment was Botanicard, the liquid formulation of Botanicard, followed by the mineral oil Safoil X. Another thing that we assessed was the actual mortality, not only how many thrips were dislodged from the cuttings, but how many thrips died. And in this case, we saw that the most efficacious product was Safoil X, followed by Velifer. Just some summary. We saw that the deep treatment with Botanicard ES and Safoil X dislodged and killed up to 100% of the second instar larvae that were placed on these cuttings. We also observed that the plant should not have thrips damage when dipped into oils, because if they do, we might see some phytotoxicity. Powder formulations were less phytotoxic than oil formulations, but overall these rates were tested on Mandevilla and Gardenia cuttings, and we didn't observe significant phytotoxicity that would prevent the health, the establishment of a healthy plant. And with that, I'm going to switch gears to biological control and update you on the most recent updates that we have on the efficacy of formulated entomopathogenic fungi. We tested different wettable powder formulations of Isaria fumosorea, Bavaria bassiana strains, but also liquid formulations of Metarhizium bruneum, again, different Bavaria bassiana strains. And we did a recipe. We followed like a protocol that is standardized, and in order to be able to narrow down the list of treatments that we want to further explore. So all these formulated products have the entomopathogens, and they come in different numbers of spores. But to give them an equal chance and equal opportunity to see their efficacy in the lab, we fixed the number of spores to 10 in the power of 7 per milliliter. So pretty much we had a homogeneous rate for all these formulated products, and this is what we assessed. And all this was done in the lab. Again, we used lifties, only this time they were much larger. And we worked again with the 3 feeding stages, first and second instar larvae and the adults. 2 types of treatments as in the chemical control. We had 10 replicates per treatment. We used the spray pot or tower to apply our treatments, and we assessed mortality at 3, 4, and 5 days. And in the case of the adults, we waited until the 7th day as well. We also test the spore viability to make sure that the product that we were evaluating had good viable spores, and we did that for all the tested products. I can tell you now that all the products had 100% viability of the spores. I'm going to show you the wettable powders, and I'm going to be showing you a series of graphs that look like this. So we're gonna go stage by stage of thrips, and we're gonna start with a direct treatment application. So when the thrips came into direct contact with the treatment, we're gonna see the proportion of dead thrips over the different treatments. And here we had a significant effect of the treatment, and also we had a significant effect of time. So if we talk about the treatments, we can see that Isarid and Ancora were significantly more efficacious than the water control. The same goes for Bioworks, the wettable powder. Um, but we had a principle and bioseries that did not differ from the water control. Another aspect is that as the time was passing, so from day 3 to 5, we saw that the mortality of the thrips would increase. Then, the proportion of dead thrips of second instar larvae thrips. Similar results. Again, the most efficacious products, actually all of them were more efficacious than the water control, but the one that stood out was Isarid, followed by Ancora. The water control, as you can see, is has significantly lower mortality in comparison to all the treatments. And again, as the time passed by, we had more second instar larvae dying. When we took a closer look on the adult mortality this time, similar results in the sense that the more time was passing, we had more adults dying because of the treatment. And again, Isarid, Ancora, and Bioseries were significantly different than the water control. They caused significantly higher mortality than the water control. When we assess the residue toxicity, so when we spray the leaf and then with the entomopathogen, and then we release the thrips larvae, we obtain similar results. So Isarid and Ancora, again, showed really the highest efficacy of all the products. And this was consistent also when we assessed the mortality in the second instar larvae. The adult mortality also was significantly higher than the water control in Isarid, Ancora, and Bioseries. And again, the same pattern. The longer we expose the insects to the treatment, the higher the mortality that we would score. We did the same evaluations assessing liquid formulations of entomopathogens. And I'm going to show you again similar graphs, only that this time the treatments have changed and we have the liquid formulations. So in the case of the first instar larvae that came into direct contact with the treatment, we can see that Lalgard, Velifer, Botanigard, and Principal were significantly higher mortality than the water control. But Lalgard really stood out. Similarly, with the second instar mortality, Lalgard and Velifer stood out and had significantly higher mortality than the water control, but also we saw that Botanigard and Principal had good efficacy. In the case of adult mortality, things were very clear already within the first 4 days. Lalgard and Velifer were the most efficacious treatments, followed by Principal and the Botanigard, and all of them were caused significantly higher mortality than the water control. When we looked at the residue toxicity, we observed similar patterns. Again, Lalgard followed by Velifer, caused significantly higher mortality in comparison to the water control. And this result was also consistent when we assessed the mortality of the second instar larvae. And in the case of adults, we had 3 treatments that really stood out: Velifer, Lalgard, and Principal. All 3 of them caused significantly higher mortality than the water control. And of course, again, the longer we assessed Thrips mortality, the higher the mortality that we would score. So as you can understand, we had a lot of good treatments, but we need to make a choice of what we are going to further evaluate under greenhouse using plants in a more realistic environment. And due to the lack of space, we had to narrow our list into the best 2 wettable powder formulations and the best 2 liquid formulations based on our lab essays. We chose 2 formulations of Isaria fumosorea, Isarit, and Ancora, and the liquid formulations was Metarhizium bruneum and a formulation of Bavaria bassiana. Something that I need to mention, we used the rates that they were recommended by the label this time. We worked with gardenia plants, the cultivar Revynthi, and what we did, we infest the gardenia plants with 32nd instar and 30 adult thrips females. We let the thrips establish for 2 weeks to really infest the plant. And what we did, we started sampling after we applied the treatment. We started sampling 5, 7, and 10 days later. And we collected leaves from the different strata of the plants, from the top part, the middle, and the bottom. And every time that we did this experiment, we had 10 replicates per treatment, and we repeated this experiment twice. So we had in total 20 replicates per treatment. So this graph shows you the population dynamics of the thrips. So how many live thrips we would score, we would find during the entire experiment on the plants that they received a different treatment. Water was our control, and you can see it with orange. And then this graph, it shows you the average number of thrips over the course of the experiment. And what you can see is that throughout the experiment, and especially towards the end, it is very evident that all the treatments had significantly lower number of live thrips on the plant in comparison to the water control. So we did only 1 application because we wanted to see 1 application, what effect was going to have on the thrips population. And even with 1 application, we could see that the number of live thrips observed on the plants, it was significantly lower than the water control. But because we were doing the samplings, we could also assess mortality. So every time we would score the live and the dead thrips. And when we assess mortality, we saw that the treatment had a significant effect, with Isarit causing significantly higher mortality than the water control and standing out. But we also saw that that the duration of the experiment of our sampling did not really show a lot of variation. So what was the take-home message was that all the treatments can actually reduce the thrips population, because we saw the thrips population being significantly lower than the water control. But when we dived into actual thrips mortality, we saw that this formulation of Isaria fungus was very efficacious. So what we can conclude is that the application type did not affect the thrips mortality. So either when it was a direct contact with the insects or a residue toxicity. When we applied wettable powders, the application type did not really vary, did not affect the thrips mortality. However, when we assessed liquid formulation, the direct contact caused higher mortality than the residue toxicity assay, because the oil itself has some properties that can increase thrips mortality. We also saw that in the wettable powders, the higher mortality was found in Isarid and Cora, right, in the lab, and for the liquid, Lalgard and Velifer, and this is why they were further assessed in the greenhouse. And in the greenhouse, we saw that all the products, even with one application, they were more efficacious than the water control. But if we want to assess overall performance and consider also the mortality, Isarid was the ones that were standing out. I hope I'm not, I still have some time. And moving very fast with entomopathogenic nematodes. We did, yes. So for the entomopathogenic nematodes, we try to target the stages that reside in the soil, so the prepupas and the pupas. We started again in the lab assessing different entomopathogenic nematodes that they are available in the market. You can see the species here. That is Heterorhabditis bacteriophora, Heterorhabditis indica, Stenennerma carpocapse, Stenennerma kraussei, Stenennerma felti, Stenennerma rio brave, and we used water as our control. In this picture, you can also see the experimental unit that we use. So we try to have soil in our assays to really assess the efficacy of these nematodes. The rate that we used was 200 infecting juveniles per square centimeters. And what we were doing is we had a lift disc that we inserted on this arena, and it has the thrips larvae. And we let the second instar larvae leave the disc and pupate in the soil for 6 days. Then we retrieved the leaf disc, and after that we inoculated the soil with the entomopathogenic nematodes. And we measured the efficacy as an expression of the number of surviving adult thrips that will emerge from the soil. Okay, so this graph shows you how many adult thrips were recovered from emerging from the soil when we applied the different entomopathogenic nematodes. We have the mean proportion of recovered adult thrips over the different treatments that we apply. And as you can see, Heterorhabditis bacteriophora, Stenennerma rio brave, Heterorhabditis indica, and Stenennerma carpocapse had significantly less number of thrips emerging from the soil in comparison to the water control. So these 4 species were further selected for greenhouse evaluations. What we did greenhouse experiments using Mandevilla plants. The cultivar was scarlet. We inoculated the plants with 50-second instar larvae, and we waited 3 days so that the instar larvae have enough time to move to the soil for pupation. And after 3 days, we applied the nematodes at a rate of 100 infected juveniles per square centimeter. And the treatments were the most efficacious, the most promising nematodes that we saw from the lab. And of course, water was our control. What we did, after we applied the treatment, 24 hours later, we installed also a yellow sticky card in order to be able to trap the emerging adult thrips. We did a foliage sampling 8 days after we applied the treatment. We cut the entire foliage and actually scored all the thrips that we can find on the flowers, on the leaves. We also replace the card twice in order to capture whatever individuals didn't have enough time to reach adulthood. So we kept the experiment going for at least 13 days. And we had 10 replicates per treatment. We repeated the experiment twice. So overall, our results show that the average proportion of recovered adult thrips was significantly lower in all the four species of nematodes that we assessed when we compare them to the water control. However, Stenainerma rheobrava, Stenainerma carpocapsae really showed very good performance in comparison to the Heterorhabditis species. So in conclusion, we saw that these two Stenainerma species show the greater potential to control the short spine thrips. And maybe combining entomopathogenic nematodes with other biological control agents has the potential to enhance the pest control effectiveness. So we have a series of resources. I have put the QR code where we are going to upload the recording, the second part of the recording of this webinar. We have published 3 papers already. If you want to get more details into our experimental procedures. And with that, I would like to thank all the funding agencies and the industry support that we have received. Of course, the Thrips parvispinus task force, the FNGLA, all the Miami-Dade County growers and stakeholders that have been supporting this research, as well as biological and chemical control industry partners. And of course, thank you for your attention. We can now move on to Paola. Paola is going to talk about the potential of predatory mites to control the short spine thrips. Paola, you are muted.
Okay, now it's fine. You can see my screen.
Perfect.
Okay, so good morning to everyone. Yes, as Dr. Revynthi say, I'm Paola Villamarin. I'm PhD student in Ornamental and Acarology Lab. And I'm going to share with you part of my PhD research. This part is the biological control of the short spine thrips, the Thrips parvispinus, using predatory mites. So, as a short introduction, why did I choose to test predatory mice? This is because we already have research evidence that for other important thrips species in agriculture, so the one showing in this slide, there are very good potential predators that can be used with very effective results. Against this, some of these species are invasive here in the United States, other in Europe, and for all of them, we have very good predators from the mites group. So the main objective of this research was to evaluate commercially available and naturally occurring predatory mites against the short spine thrips. The Thrips parvispinus are here. There is the list of the predatory mites that I tested for this evaluation. These are the commercially available predatory mites here in the United States. All of them belong to the Phytoceae family, with the exception of Anistis baccharum, that it belongs to the Anistidae family and is, let's say, is bigger in size and also in behavior, as I will explain later. And also Stratylolera simitus, that is a soil predatory mite. So we found them especially in the soil. And I also test 2 naturally occurring predatory mites here in South Florida, Propioceapsis ovatus and Amblyseius largoensis. So this research was divided into parts. The first part was a choice experiment. And with this one, we want to answer the question if can predators prey on the short spine thrips and which state do they prefer. So this was a choice experiment. And To explain a little what did I do, this is the methodology. I use these petri dishes or these arenas that I'm showing here in the blue tray with a bean leaf disc on top of a wet cotton wool. On each arena, I release one female of each predatory mite with a combination of two Thrips stages, especially first and second instant larvae, but also in some cases I include prepupae, pupae, and adult in some special cases that I will explain through the presentation. The evaluation, the time of the evaluation of this part of the experiment was every 5 minutes for the first 30 minutes, every hour for the next 6 hours, and 24 hours after the release of the predatory mite on each experimental arena. This experiment was has 8 replicates and was repeated three times. And the response variable that I was measuring in all this evaluation time was the mean proportion of the predatory mites that make a choice for one of the offer stages according to the treatment. So to talk a little about the results of this part of the experiment, I will show you a series of graphs like the one here in the slide. This graph represents the mean proportion of the predatory mites that make a choice for one of the offer stages. So in this case, in the left side of the y-axis, we have the predatory mites that make a choice for first instar larvae in the bar in blue color, and in the right side should be a bar in orange color if the predatory might make a choice for second instar larvae, as in this case. In the bottom of the slides, I will show you what was the first choice and the time of this first choice of each predatory mite. So to start with Noceolus californicus, this predatory mite, as you can see here, make a, not most of them make a choice for first or second instar larvae, and this is because this predatory mite is mostly selective for spider mites. But we decided to test it because there is research that it can feed on other Thrips species such as Thrips palmi. But in the case of Thrips parvispinus, we can see here that there is no high preference for this Thrips species. In the case of Amblyseius swirski, when we offer first and second instar larvae, there is a clear preference for first instar larvae, with more than 60% of the predatory mites making this choice. The first choice was recorded after 5 minutes of the release of the predatory mite, and a very short proportion of these predatory mite species make a choice for second instar larvae. For this predatory mite species, I also include a prepupia stage in the combination because Amblyseius swirski we usually found foraging in the canopy of the plant, but it can also walk through the stem or in the soil or litter around the plant. So it can go also for prepupia stage, that is the first stage of Thrips parvispinus that with development on soil. So when I offered this combination of Thrips stages to Amblyseius swirski, we didn't find significant differences, and there is preference for both stages. And the first choice in this case was recorded after 5 minutes, with no difference for first or instar larvae of prepupae. When I include, when I offer to this predator species the combination of second instar larvae and prepupia stage, we can see a clear preference for prepupia stage instead of second instar larvae, and this is because second instar larvae has a stronger defensive behavior against predation than prepupia that is a less mobile stage. In this case, the first choice was recorded after 5 minutes, with making this first choice for prepupia stage. In the case of Amblydromalon legmonicus, very high preference for first instar larvae. This choice was recorded after 5 minutes of the release. More than 60% of the predatory mites made this choice, and very short proportion of the predators make a choice for second instar larvae. In the case of Noceolus cucumeris, complete preference for first instar larvae. More than 80% of these predator species choose first instar larvae, with the first choice recorded also after 5 minutes. In the case of Amblyseius andersoni, similar behavior, compared preference for first instar larvae with more than 60% of the mites making this choice. And in this case, the first choice was recorded after 10 minutes of the release. For Revynthi degenerans, that is a pollen feeder, but also very well used against Thrips pests. High preference also for first instar larvae, and a low preference also for second instar larvae. In some cases, we recorded this choice. And in this case, also the first choice was recorded after 10 minutes of the release of the predator. In the case of Anistis baccharum, when we offered first and second instar larvae, we didn't find significant differences against between these two Thrips species, but we can see here in the graph that most of the preference is for second instar larvae. And the first choice in this case was recorded also after 5 minutes of the release of the predator for second instar larvae. For this predatory mite, I also include in the combination in this choice experiment adult, because the size of the predatory mite and also the feeding behavior allows this predatory mite to also choose this stage of Thrips. So when I include the combination second instar larvae and adult, we also can see that this predatory mite can feed on both stages with no significant differences, and very high proportion of the mite exhibit feeding behavior on both. And the first choice was recorded after 5 minutes, still with the first preference for second instar larvae. In the case of the soil of predatory mite, when I include also in the combination for this predatory mite second instar larvae, despite this stage is mostly developing in the canopy of the plant, it goes to the soil before to become prepupil. So there is a time that the predatory mite can have a space to feed also in this stage. So when I offer these two stages to this soil predatory mite, we didn't find significant differences, and it can feed on both, with a good proportion of the predatory mite making the choice, and the first choice was recorded after 5 minutes of the release. When I offered this predatory mite with second instar larvae and pupae, we also didn't find significant differences. The mite exhibiting feeding on both stages, and the first choice was recorded also after 5 minutes, with no difference for what stage. And when I offered this predatory mite with the two stages with development on soil, we also we found that it can feed on both stages with a higher preference for prepupae stage. And in this case, the first choice was recorded after 5 minutes of the release of the predatory mite in the arena. Regarding the naturally occurring predatory mite Proposiopsis ovatus, when I offer larvae stages, it showed a small preference for first instar larvae. Around 20% of the mites make this choice, and the first choice was recorded after 10 minutes of the release. This predatory mite exhibit similar feeding behavior as Amblyseius swirski. It can walk through the stem and can be around the litter or soil around the plant. So I also include combination with prepupae stage, and we can see here that also this predatory mite can feed on this soil stage of the Thrips with no significant differences when I offer against first instar larvae, and the first choice was recorded after 5 minutes for prepupae stage. When I include second instar larvae in this combination, because due to the behavior, defensive behavior of the second instar larvae, as I explained before, the predatory might choose more prepupae stage, with the first choice recorded after 5 minutes of the release. So from the first list that I showed you at the beginning, there are predators that do not feed on Thrips parvispinus, and these predators are the naturally occurring predatory mite Amblyseius largoensis and also Neoceruleus longispinosus, because this is a predatory mite also mostly selective for spider mites, but we decided to evaluate because there are research that they can feed on other Thrips species, but it's not the case for Thrips parvispinus. So as a small conclusion for this first part of the research, Most of the phytosedemites can feed prefer a first instar larvae of Thrips parvispinus. Also, in the case of Amblyceus swirsky, it can feed on prepupy stages. In the case of the predatory mite Anistis baccharum, it can feed or it prefers most second instar larvae and also adult stage. That is something very interesting. The soil predatory mite can feed on the Thrips stages with development on soil and also second instar larvae. And for the naturally occurring Propeociopsis ovatus, that was the only with exhibit some preference for Thrips stages, it can feed also in prepupae and first instar larvae. So as a follow-up for this first experiment, I conduct a no-choice experiment to answer how many short spine Thrips larvae can the predators consume and how many eggs do they lay. Of course, this experiment includes just the predators that exhibit some preference for Thrips parvispinus. In this case, I use the same arena as before, with the difference that I release one female predatory mite of the mice with preference for Thrips parvispinus and a first instar larvae. And the evaluation for this experiment was every 24 hours for 4 days. In this evaluation time, I evaluated the number of consumed larvae, the number of live larvae, and also the number of predator eggs as an important measure of the reproduction of the predators after feeding on Thrips parvispinus. This experiment included 10 replicates per treatment, and it was repeated 3 times. So to start talking about the results, this part about the predation of the predators on the short spine Thrips, I will show you this graph that includes in the y-axis the average consumed larvae per day, how many larvae do the predators consume, and the x-axis all the predators that I test. So in this case, for Amblydomados monicus, we found that it can consume around between 3 and 4 larvae per day. Amblyceus andersoni, a little less, can consume between 2 and 3 larvae per day. Amblyceus swirsky, more than 3 larvae per day. Epiphyseus degenerans, the highest value of predation, between 5 and 6 larvae per day. In the case of Noceulus cucumeris, similar to Limonicus, between 3 and 4 larvae per day. The natural occurring Propeociopsis ovatus, between 1 and 2 larvae per day. And for the soil predatory mite, we decided to evaluate predation against second instar larvae. That was one of the most preferred, and also the prepupy stage. And we found an average consumption of more than 5 second instar larvae per day and more than 3 prepupy stages per day. So regarding oviposition, I will show you the same graph, but in the y-axis, I will show the average predator eggs per day. So in the case of Amblydomados limonicus, it laid between 0.2 and 0.14 eggs per day after feed on Thrips parvispinus. Amblyceus andersoni, higher than Amblydomados limonicus. In the case of Amblyceus swirsky, more than 1 egg per day. Epiphyseus degenerans, similar to Amblyceus swirsky, with more than 0.8 eggs per day. Noceulus cucumeris, similar than swirsky and degenerans. The naturally occurring predatory mite, the lowest oviposition rate, less than 0.2 predator eggs per day. And in the case of the soil predatory mite, when feeding on second instar larvae, more than 0.6 eggs per day. And when feed on prepupy stage, similar to the naturally occurring predatory mite, around 0.2 eggs per day. So for the case of Anistis baccharum, I separate the evaluation for this predatory mite because it has a different feeding behavior, more voracious, I can say. So in this case, for Anistis baccharum, I offer second instar larvae instead of first instar larvae, and I offer 15. So I made the evaluation similar as for the phytosedemites, and in this case, we found that it can feed on more than 12 second instar larvae per day. Also, in the case of oviposition, This predatory mite is very different also in biology. The position is a mass of X of around 24X, and we found this mass of X after Anistis bacarum feeding on second instant larvae of Thrips parvispinus. So as conclusion, for most of the predators, they prefer first instant larvae of the short spine thrips, especially the phytosaide predatory mites, the commercial predatory mite Amblysserius swirsky, and also the natural occurring Propiosiopsis ovatus can prey also on prepubic stage. In the case of the predator Anistis bacarum, interestingly, it can feed also in adults. And in general, Amblysserius swirsky, Iphiseus degenerans, Nesuiolus cucumeris, Amblydromalus limonicus, Anistis bacarum, and Streptococcus lacimitus show highest potential as biocontrol agents against the short spine thrips. And the use of these predators are especially important because all of them are generally predatory mites, so they can be released before the pests arrive, and we can create a standing army to be ready to control the pest when it arrives. So the ongoing research about this part is the evaluation of these potential predatory mites on Mandevilla and gardenia plants, and this is conducted on greenhouse conditions. So with this, I finish my presentation. Thank you to my lab members, also to the companies that provide us with the predatory mice, to the funding agencies, and thank you all for your attention.
Thank you, Paola. Is Livia here? We are gonna continue with the potential of predator insects against the short spine thrips.
Yes, I'm here, Alex. I'm going to share my screen. Is it okay? Can you all see my screen?
Yes, perfect.
Okay, great. Okay, so thank you very much for this opportunity to be here today to talk a little bit about predatory insects in the control of the short spine thrips. And as Alex already mentioned, I will be showing you some results about the efficacious ability, the voracious ability of these predators towards the short spine thrips, and some of the results that we are also some of the experiments that we are going to perform in greenhouse. So I'm sorry, I'm just having a problem here trying to change my slide. Sorry, just give me a minute.
We can see the slide without any issue.
Yeah, I'm just not able to change it. Just
just press on the screen.
It's just because I think I'm trying to hide the bar that I can see here. Yeah, now it's okay. Okay, now it worked. Yes. Okay, so I will focus on 4 natural enemies here today, 4 predators. And the first one is the pirate bug, which is this one here that you see feeding on the thrips. And as you can see, it's way bigger than the predatory mites that Paola was showing. And in this case here, you see that it's still small, but it's at least 5 times bigger than the thrips. So the pirate bugs, they are actually voracious natural enemies against thrips. And we tested if it was also a voracious predator towards the short spine thrips. Another natural enemy here that I tested were the mired bugs. Actually, I tested 2 species, and as you can see, they are slightly bigger than Orioles. The next one is the green lacewing, and in this case here, they feed also on several other insects. And you see here in the photo that they are feeding on an aphid. And then the last one was the rope beetle. And these are actually soil predators. They are very small. And as Alex mentioned, the thrips, when it's about to pupate, the larvae move to the soil and then they pupate in the soil. So the target stage for this rove beetle
Lydia,
here, for this, yes.
Lydia, I'm sorry, can you increase a little the volume? Because people are not able to hear you well.
Yeah, well, how I'm gonna do that?
And try also with your headphones.
Maybe I can try to remove it. Okay, let me try to remove the headphone. Is it better now?
Yes, Lydia, thank you.
Okay, I'm sorry. Okay, so let me continue from here. So the first one that I tested was the pirate bug. And as you can see here, it feeds not only on thrips, but also on aphids, caterpillars, whiteflies, and other soft body insects. So it was already a very good candidate that we test for the short spine thrips. And just to give you a little bit of information about its life cycle, so after the female laid eggs, it actually laid eggs inside of the plant tissue. And once the egg hatch, you have 5 wingless nymph stages before it reaches the adult stage. And the whole cycle takes around 21 days. So the stage that I tested here was the adult stage. So I used adult stage females of the pirate bug, and I checked how many thrips, larvae, and adults the pirate bug could feed per day. So what I did here was a non-choice experiment, and I used this setup that you can see here in this photo. So as you can see, you have here agar and a leaf disc, and then I placed the thrips first, and depending on the stage, so it was just L1 or L2 or adult thrips, and then I placed the predator in the arena, and I close the pot, and I wait for 24 hours. After 24 hours, I counted the number of eggs and the number of carcasses that we found in the leaf discs, on the leaf disc, sorry. So what I'm gonna show here is the number of dead thrips that we found after 24 hours. So here in this figure, you can see that I offered 30 L1 larvae during only 2 hours, and we could already see that they were killing quite some thrips already. And it was actually very difficult because they were so voracious to give them enough larvae to eat for 24 hours. So in this case here, I'm not going to show you the results for 24 hours because we were actually not able. We were offering hundreds of larvae, and they were actually, or larvae was escaping, or they were able to prey in all of them. So, but for larvae 2 and for adults, we offered 110 larvae 2. In this figure here, you can see the number of dead prey that we found. The number of dead prey is always shown here in orange. And here, what you can see in gray is the control. So that means that in that vials, we didn't add the predator. And in all cases, you can see a big difference, meaning that the number of dead Thrips parvispinus that we found, which we verified by the number of carcasses that we found on the leaf disc arena, it was higher than on the control treatment. In the control treatment, we have a very low mortality, but when we added the predator, we could really verify this information. So it was really interesting to also see that the pirate bug also laid eggs after feeding on thrips. This is also very important, right? Because the predator needs to eat it and be able to develop also on that prey. And the next predator insect that we tested against short spine thrips was the mirrored bug. And here, as I mentioned, I tested 2 different species. And it's actually also a predator that can feed on whiteflies, spider mites, aphids, other thrips species and leaf miners. And you can also see here that these two species, they differ in color, so it's actually quite easy to distinguish them. But one important information to mention here is that Disiphus hesperus is sold by companies, so it's commercially available. Growers can buy it, while Macrolophus preclarus here is present in Florida. It's a native predator that you can find here, but it's not commercially available. But since we had it here, so we started a colony in the lab, and we started testing this predator here as well. So we tested both predators. Here is the life cycle of the mirrored bug, similar to Oreos, similar to the parrot bug. They also lay their eggs inside of the plant tissue, and we have 5 nymphal stages before they reach the adult stage. And something important to mention here is that the life cycle of the mirrored bug is highly dependent on temperature. Under optimal temperature, they will have a shorter life cycle. But when, for example, the temperature is around 59 degrees Fahrenheit, you can have a life cycle that can take around even 90 days. So you see that it can interfere a lot in the time that they need, the developmental time that they need to complete from egg to adult. So first I'm going to show you the results for Macrolophus preclarus. So we did the same as we did for the pirate bug, non-choice experiments offering larvae 1, larvae 2, or adults in the arena, and we checked the number of carcasses, the number of dead thrips at the end, and we measured oviposition. So here are the same similar figures, right, in which you see here in orange the predation, and here in gray the control. So in this case here we didn't add predator, in this case here we add predator. And in all cases you see a difference between the control and the treatment, meaning that the predator actually fed on the short spine thrips, and but in this case here we didn't observe eggs in any of the treatments. The next one is the Disiphus hesperus. So it's the other mirrored bug that I showed you the picture previously. We did use the same setup, counted the number of carcasses, and checked if there were eggs on the arenas, and this is what we obtained as a result. So again, orange is are the petri dishes that we added the predator, and gray the control, no predator. And it's interesting to note here that they don't seem to like adult thrips because they didn't really show mortality. Here in this case, we didn't see many carcasses, so they don't seem to really prey and like to prey on adult thrips. And we also didn't observe predatory eggs in any of these treatments. The next one is the green lace wings, and they are also, they can prey on several insects as well, and thrips is one of them, so that's why we tested it against the short-spined thrips. And it's commercially available as well here in the US, and it's interesting to show you here that the nymphs are predators, but the adults, they are not. They feed on pollen and nectar. Okay, so, and they looked very different from each other. That's why they are called green lace wings. But the larvae, as you can see here, is brown, it's not green. And it's also interesting to see how they lay their eggs. So this picture that you see here, it's a silk with the egg on top. So this is how they lay their eggs. The life cycle of the green lace wings is, it has some larval stage, in this case here, 3 larval stages, and it pupates. And when they pupate, they make this, see the silk around the pupae. They usually pupate in hiding places. We can be also in the soil, or they look for places that they can stay hiding from predators or environmental factors that might affect them. And then they become adults and they lay eggs. In this case here, it's also important to note that they are also highly dependent on the temperature.
to complete its life cycle. So the green lacewings also showed that they are highly voracious against the short spine thrips. We can see here that they also didn't like to prey on adults too much. And it's also interesting to see here that the stage that I test for the larvae here, for the green lacewing, was the second stage. So they were a little bigger, not so small. And in this case here, we see that they seem to like to prey more the larvae too. And the reason why I believe that is just because they are bigger than the larvae one. So they also like to prey more the bigger thrips than the small ones. And we could see that there was like a preference towards the L2 stage than the L1 stage. But I didn't test preference here. Here is just non-choice experiments, but it's just something that I wanted to share with you about what we saw it during the experiments. And here you can see what I'm saying. So here you see a larvae, and the larvae here grabbed an L2 thrips. And you can see, you are gonna see that soon is gonna, some L1 larvae are gonna cross here, but they are too small. Look, this is larvae, a smaller larvae, and they are too small, so they seems to be ignored by the green lacewing larvae. And they are very voracious, so they have these pincers. They used to grab it and also to suck up the content of the thrips, and they can feed it in a matter of seconds. And here you see a carcass that this guy here already fed. So you see it's getting emptier and emptier, and soon it's just gonna be a carcass on the leaf. So the last one that we test was the rove beetle. As I mentioned, they live in the soil, so they prey small insects that live in the soil. And it actually has been used by producers, by growers here in the US against fungus gnats. It's one of its favorite prey. The life cycle of the rove beetle also has a larvae stage and is quite fast compared to the other ones. It takes around 17 days under optimal conditions. And so you have the egg, the larvae, the pupae, and the adult stage. So how did we test it? In this case here, we had to change the setup. So we could not use the leaf discs anymore because they are soil predators. So we used a similar setup that Alex mentioned during her presentation when she showed the nematode data. So we had a vial, we had to put soil, and we added then a leaf disc with 10 thrips larvae. We closed the pot and we waited for a couple of days. And then what happened was that the larva is gonna move to the soil, the thrips larva is gonna move to the soil to pupate. And therefore, the predators were added a few days later when we saw that there was no larvae on the leaf disc, which means that the larvae migrated to the soil. We removed the leaf disc and then we place the rove beetle. And we place only one rove beetle per pot. And then we close it and we placed this yellow sticky trap that you can see here on top, because the goal here was to count how many adults you would see trapped in the yellow stick trap. And we would consider the number of missing adults out of the 10 thrips pupae that were offered as a predation rate for this guy here. So the number of missing adults, they were considered preyed by the rove beetle. And this is what we obtained. I did actually many treatments, many replicates, and because I was really trying to understand better if this predator was a good predator or not for thrips pupa. But at the end, after more than 40 replicates and in 3 different blocks, we saw that they don't seem really to prey and like to prey too much on the short spine thrips, okay? Because there was no difference between vials in which we added the predator and vials in which we didn't add the predator. So as a conclusion here, we could verify that indeed in the lab experiments, the pirate bug seems to be the most voracious predator, that insect that we have so far. But the mirrored bug can also feed on the short spined thrips, but they don't lay eggs. While the grace-winged larvae, they proved also to be a voracious predator of the short spined thrips. But they like the larvae, they didn't like the adults. So as you can see, they have some advantage and disadvantage when you tested all life stages and when you check their oviposition and their predation. They have difference because they are different organisms, right? Which was already expected. And the rove beetles, they did not seem to like thrips, at least these thrips, the short spine thrips, too much. And at the moment, what we are doing, we are carrying out experiments in the greenhouse, testing the top candidate, which is of course the pirate bug and the green lacewing. But we also included the mirrored bug, the Disiphus hesperus, which is the one that's commercially available, because we know that this one is quite promising as well. But these experiments, they are being currently, they are in progress. Okay, so this is what I have to tell you today. I really hope you have enjoyed the presentation, and I thank you very much for your attention. Thank you.
Thank you, Livia. Right on time. So the last talk of today is going to be from Dr. Lance Osborne, who's going to be talking about control of this pest, but in the greenhouse. So we're going to see a different perspective. Lance, you are muted.
Always one person that forgets to turn on his microphone. Okay, I appreciate the opportunity to be here. I have a number of people to thank that helped me with this research. Some of it has been done by Dr. Park, a postdoc in my lab, Catherine Hubin, Michael Hill, and Cindy McKenzie. That's not getting off to a great start here. Okay. Obviously, with thrips management, it starts with scouting, trying to determine the population densities, when you can apply certain things. Is just absolutely critical knowing what you're dealing with, especially when you're dealing with biological control agents. You need to be able to identify them and follow what's going on with their populations. With the thrips, the first sign that you're having a problem, obviously, is the damage, because these things are so small. Damage has probably gone way too far, but it's Asclepius and Hoya showing significant damage from this particular pest. We recommend that you spend some money to get good hand lenses. In this case, they're Bosch and Lomb 10x and a 20x. We use ones that that are going to cost $30 to $50 a piece, but they're definitely better than the little plastic ones that you get handed in a trade show. And you know, if you're going to be doing this, you need to get the right tools to do it. Okay, this is parvispinus. As everybody's seen, we'll go through this fairly quickly. Just adult female. And the problem that we do have a little bit here at my research center is that the males look just like chili thrips. So when we're counting, it's hard to tell whether we're dealing with a mixed population of species or whether it's just parvispinus. Routine scouting, we recommend using a beat board, in this case just a clipboard with white paper. Sometimes we can use a paint easel, and it's a little bit easier to deal with and easier to clean. This is my biologist sampling for thrips and roses. It works well, but the bottom line is that it's hard. You try to get your head down there to see what you're dealing with. Obviously, she's probably filling her hair with more thrips than are on the board. But it's something that we've been working with. With thrips, it's a lot more difficult because they're small, they're very active, and by the time you get down in there, they've all scattered away. We took to putting sticky cards on plates so that when we beat them against the plate, we could find and count a certain number of the pests. We thought that the sticky cards would prevent the spread of the movement of the pests off of the plate, but we found that this particular color, the yellow ones, were not that sticky, and parvispinus would get up and walk off. So you have to evaluate your tools to make sure that they are doing what you want them and expect them to do. So this also puts if you're using the cards to monitor populations out in the greenhouse, you may be getting erroneous data if you're using the yellow sticky cards here. Not all yellow sticky cards have this issue, so you need to check what you are using. The blue sticky cards had a different stickum on it, and it appeared to be a lot more sticky, and it would catch the parvispinus, and it wouldn't get up and walk away. So there are other tools. You're going to find a lot of different thrips when you start sampling in Florida. We have this particular guide, Pest Thrips in the United States, but it's mainly done with the thrips that we have here in Florida. So you can compare them to parvispinus and then other ones that you might encounter. Chemical management, Alexandra, Dr. Revynthi did an excellent job covering most of that, so I'm going to skip chemical control. Pesticide-induced pests. Now, this is something that you know, if you're monitoring, you need to really pay attention to. We've learned the hard way that this can be an issue, and it points out one thing that we don't want to do, and that's use bifenthrin or Telstar to manage these particular pests. The father of biocontrol basically stated in one of his mantras was when we kill off the natural enemies of a pest, we inherit their work, which is kind of what we're doing now. In a chemical evaluation efficacy trial with a number of different treatments, we had obviously a water control, and then you can see that we had significant damage to this pepper as a result of the thrips. We had one treatment in there that was treating with Telstar, and you can see that the plants are in worse shape than the control plants were. We're not sure exactly what's going on here, except there's a phenomenon called the hormolygosis, where when an insect has been treated, it appears that they start laying more eggs as a result of the chemical application. So we ended up with more thrips on this plant than the control, and it looked like on the fruit that they were going to carry the fruit away. There were so many thrips running around on it. Now, in this case, we didn't have parvispinus. We're not sure exactly what happened. They were displaced by the chili thrips. Okay, management and biological control. There are a lot of predatory mites that have been mentioned today and that are commercially available in the U.S. You will see in the literature and some of the comments by other companies, they have different kinds of biocontrol agents, and they mention in some cases, we have a company that was talking about monitorensis, and that is a predator that's not allowed to be used in the U.S. So you need to make sure that your company is selling you what's appropriate, and I don't think anybody is selling the illegal ones. But we've had some confusion where people were trying to get these predators that they read about in the literature, and they're not available in U.S. Here's Swirsky feeding on a thrips egg. It'll feed on any stages that it can, as we've already heard. This is one of our go-to predators. But the problem in Florida is that we've had it, especially outdoors, that once we get into the fall, they didn't work as well as they did during the spring. So they're somewhat sensitive to the temperature. So again, you need, when you're buying in predatory mites, you need to discuss this with your rep as to what type of environment these things are going to be placed in, and be cognizant that they don't work quite as well during the winter months. This is not a banker plant system, but it's an open rearing system of sorts. You realize that these sachets and some of the containers, when they sell it to you, have bran in them and have a source of food for the predators, which is a stored product mite. And so you'll see a lot of things running around. Most of them are probably stored product mites if you just purchase these. And they act as a time release, basically, for the predators. They'll feed on the bran mites, and they will, over time, disperse from these little sachets. There's a little hole right here in this drawing of a mite, and that's where they exit and then move throughout the crop. So again, they can be releasing predators for a couple weeks at least, if they don't get wet. Now that's a problem we have in greenhouses where they have overhead irrigation, and these things are useless the day after they get treated with water or get sprayed, because it'll just turn to mush. So you have to protect them. Some of them say, companies say theirs are waterproof, but we've had poor results with anything if it gets wet. We hang these in the crop. Again, this is a time release. It's almost a banker plant system from the standpoint that you can put them out and have what was mentioned earlier as a standing army. The predators move out and look for things to feed on, and if parvispinus happens to show up, you already have predators in the environment to take care of them. This is basically what it looks like. You need to realize, excuse me, that these predators all have certain needs that they need to be happy, to be useful, to be doing what you want them to do in the greenhouse. They need an acceptable food source. For example, with Swirsky, they will feed on pollen, and we used to buy in pollen and supplement their food with pollen, but we can't get the pollen anymore. We're not really sure why companies quit selling it. Some of these predators will feed a little bit on fungi. They'll feed on other arthropods, spider mites a little bit, broad mites, basically Calimbula, things that they can catch. They will feed on and keep them in the environment. You can buy Artemia cysts, which are the kind of the reproductive, almost like eggs for sea monkeys or brine shrimp. You can buy those commercially, and the predators seem to really like them. There's a statement on the ones that you buy that, if you're allergic to shellfish, you have to be careful with these things. Then there's Asphestia eggs or moth eggs that are commercially available, and I believe they're cheaper than Artemia. And again, they use those moth eggs to feed minute pirate bugs. They use them to feed a lot of other predators. They need places to live, okay, and hide and lay eggs. And in this case, this is a pepper plant, and we evaluated a lot of different varieties of peppers to find ones that had good tough domatia, which is what these are called. These are little trichomes and hairs that are in the axil of the veins, and the predatory mites like living in there, and it's a place that they hide. The predator that we're going to talk about in a few minutes, the predatory thrips, will pupate in here. They also like these hairs to lay eggs on. So you can see a lot of these hairs will have eggs of the predatory mite. And this kind of tells you, it gives you a place to look for predators to see if they're active in your crop. But it tells you whether they're happy, if they like the situation that they're in, basically. So we've tried to figure out how to give our predators some of these needs, to fulfill the needs for them. So we can get, like, we use a banker plant that has pollen. It may be producing. or in the case of the sachet, it's grain. You have the stored, I don't know why this is changing on me, but stored product mites and or pollen is some of the food. You can also augment with the eggs, Svessi eggs, or cysts. You don't have to in all cases, but in some plants, this might be warranted. We have found that, for example, we can't get predatory mites to feed on thrips and roses. We have major issues until we start supplementing and putting things near them, like a banker plant that has a place for them to live and move out from there to feed and search for predators on the roses. And these types of things we use for both predatory mites, aureus, and Franklinothrips, which is a predatory thrips I'll talk about in a few minutes. These are some of the examples of what we've used. Black pearl was used up north in many of the greenhouses for, I'm not touching anything, it's just doing this. They feed on black pearl is put in the greenhouses to augment and supply aureus with the hiding places and alternate food sources. They feed on the pollen as well. And so we also looked down here in Florida at a number of different varieties of plants, and we've settled with red missile and explosive ember as a couple that work really well for us to maintain Swirsky in commercial greenhouses for thrips and augmenting control of whiteflies with the predatory mites. We have a paper that we published, Dr. Eric Scholler, Cindy McKenzie, and myself, on looking at Lamonicus and Swirsky on roses for the control of chili thrips, using basically banker plant system to maintain them in the system, and it worked very well. And this is the first time we've been able to control chili thrips on roses using predatory mites. Without the banker plant system, we were not successful. And this is an outdoor situation, and you'll see that there are some pepper flowers here. You don't even notice it, and they basically serve as a resource for the predatory mites so that they can move up into and search on the roses and manage the excuse me, the spider, the thrips there. Plus they'll feed on spider mites if they happen to find them. We also are trying to figure out whether we can augment some of the natural controls that are already out there searching the plants. In this case, a fly that's attacking thrips. They're very common. You'll see these in the long-legged flies and some of the metallic-looking flies that are sitting there and basically sitting on the edge of a blossom and diving in and feeding on the thrips that are there. This is just something that we're interested in. We haven't gotten very far. This is the aureus feeding on thrips. We find them to be very voracious and one of the better predators for all the thrips in the greenhouse. But we do have problems in the winter when they start to go into diapause. Populations begin to drop off. We have a predatory thrips that we've been evaluating, Franklinothrips vesciformis, which is indigenous in Florida, and it is very effective. We found it will control a number of different thrips in the greenhouse, but they're particularly good at managing Echinothrips americanus, and this is one that has the people in Europe working on trying to find a method to manage that particular thrips because it has a significant anti-feeding or anti-predator reactions to the predatory mites. Predatory mites don't seem to work well, where Franklinothrips does an excellent job, and it can turn around a population and knock the thrips population down very rapidly. The problem is that it's not commercially available. They master it and sell it in Europe. They would like to sell it into the United States, but we're working with them and developing the genetic tools to separate Thrips species and to demonstrate that all the populations that we're able to get our hands on from all over the world, that they're all almost completely identical. So that means that USDA APHIS will more than likely allow us to bring them in because we are not bringing in something that we don't already have. And so the major company is in the process of getting the regulatory approval to start selling them into the United States. They feed on thrips, they feed on whiteflies, they feed on spider mites. So they're very voracious. Maybe not at the level of Aureus, but for thrips control, they've been quite good for us, with the exception of sometimes we have failures with parvispinus. And I think that has to do more with the host plant that we're trying to get them to work on, because they do lay their eggs in into the plant. And we think that maybe there's some sort of, depending on if it has a latex sap, that might have an impact on the eggs. This is the adult stage. This is the immature stage. They look like small ants running around. And again, the bright colored larvae are very easy to spot. This is just to remind me about the I didn't get that right. Anti-predator behavior. Some of these thrips where they'll flick fecal material at the predator, or they'll just bump them with the back end of their abdomen, and it causes, in some cases, predatory mites to just take off and leave them alone. And there's a number of papers out on this, and that parvispinus does have, maybe not as much as this one, but like other, it has that behavior, and it could be impacting some of our natural enemies. Okay, this thrips is so aggressive that it doesn't seem to be impacted by this anti-predator behavior, which is shown quite a bit in by the echinothrips, and it is not phased by echinothrips. It'll feed on any stage it can catch. So we know that this thrips is predatory thrips is effective at controlling chili thrips and echinothrips americanus, and we have one study that's showing that it is very effective in cages on Asclepius. Okay, so we're looking at one of the plants we've had problems with predatory mites, and that's Asclepius. It's a work in progress. Have finished one rep, and we're repping these through time. So we've got about 3 more experiments planned. But so far, so good. It looks like we have the ability to manage this thrips on one of its preferred hosts. Now, in talking to the company that's trying to market this in the United States, they said they have limited field or laboratory data on this particular predator. They say the growers predominantly use A. Swirsky and the predatory mite alongside Franklinothrips and Aureus, and in some cases they'll use Lamonicus as well. They rely on Franklinothrips more to clean up a hotspot. Because these things are, again, so aggressive and they will eat so many thrips, and use the predatory mites and Aureus as a general blanket treatment, similar to what they did for Western flower thrips. Okay, we have Dr. Park has been looking at the functional response of this predator and of Swirsky to the Thrips parvispinus. I'm not going to go into any great detail, but we rear the parvispinus on the lima bean cotyledons. In the greenhouse, we mass rear the vesperformis just in the corner of the greenhouse with beans and that are infested with two-spotted spider mite or echinothrips. And we can get large numbers of thrips just in this one little setup for doing our research. Swirsky, we rear them on the brine shrimp. And basically, to evaluate these things and their predatory ability, we take similar to what Dr. Revynthi's group has been doing, and we put them on a beans disc, and we put various densities. We release one second instar larva of Franklinothrix, and then we monitor for 24 hours and to see how many they feed on. And then we do the same thing with Swirsky and try to get the same number of individuals per dish and see how many they feed on in a 24-hour period. Basically, and then there's some math involved, and it gives you some data as far as the feeding curve. But for our purposes, basically, you can see that you give them one. This is a Swirsky. In general, they'll eat a half of one in a 24-hour period. Okay. As you increase the number, they obviously increase the numbers that they feed on. With Vesperformis, you can see here they basically clean it up until they get, until they get to such large, 10. If there's 10 in there, they'll feed on 2 to 2.5. Whereas with the Franklinothrix, they'll feed on about 7. So it's basically just showing you they're more voracious and gives you some idea and some way to compare one predator to another. So these things are better suited for managing thrips at higher populations. But they can and will lay eggs if given them just 1 or 2 thrips per day. Now, these systems aren't acting in a vacuum, so trying to use predatory mites or the predatory thrips, we found that we soon had major issues with other pests coming in onto the plants that parvispinus likes to feed on. Just about everything we've used, gardenia, diplodenia, mandevilla, asclepias, hoya, they all have mealybug problems if you don't spray them. And so we're limited to trying to manage these alternate pests. They're disrupting many of our traditional IPM programs. So it's not just parvispinus, but spider mites and those types of things that the growers that are trying to use some biological controls are reporting more and more problems with mealybugs. This is Pharesia variegatus, a striped mealybug on a plant, and they will kill a plant within a fairly short amount of time. So we're trying to find ways to manage it. We know that many of the chemicals that are used and recommended for mealybugs can be very disruptive to biological control, especially with acephate and a couple of these other materials can be very disruptive. So we've looked at biocontrol. Our growers and our homeowners and the people that are buying the plants don't know the difference. And, the ladybug is cool, but when they see the larval stage, they don't want to buy the product. They complain about buying a product that has these large larvae. And so we have to find something else because these are not really being accepted. And we've had problems with ones that we have bought in that just didn't do the job, didn't lay any eggs. So we have a poor selection of natural enemies commercially that we can buy for mealybug control. We have some cases where we've had poor efficacy, poor acceptance by the general public. And again, some of them have had some issues with maybe being infected with problems themselves, and thus not laying eggs. We have this one little beetle here that's, you can see it next to a mealybug called Diomus austrinus, which we're using for mealybug control in combination with parasitoids and getting very good results. Here's the larval stage. This is the larval stage feeding on a full adult mealybug, and it's removing the fluids. Again, this is the mealybug, and this is the beetle. So they're very tiny, and we don't have the problems with people objecting. We have a number of parasitoids, especially for Phreesia, that we're looking at in combination. And we're trying to figure out ways to get them into people's greenhouses or find commercial people that are willing to try mass rearing these. And we'll skip over that. What else? We find that these crops are also having problems with aphids. And again, we're having to find other biological control agents that can be integrated with our Thrips biocontrol. We've also found in greenhouses ghost ants. Where we have ghost ants and another ant foraging, we don't have problems with thrips, especially parvispinus. Parvispinus is very, very skittish, and if you go to try counting them, they run away, and they seem to respond to the ants foraging on their plant by taking off. And we have greenhouses where we have ants present, and we really don't have any problems with parvispinus. We published one paper on there. They do control spider mites, ghost ants do. We're waiting for another one to be accepted where it was controlling echinothrips. And we've, like I said, observed significant reduction in infestations and damage caused by parvispinus when ants were present and foraging on the plants. We're also looking at spiders. They've come into our greenhouses, and where we run into these spiders, we have problems running efficacy trials or with either biocontrol or chemicals because of these spiders. Just shots of what the little tiny ones look like. This is an adult feeding on a parvispinus. We also are finding these things are also attacking two-spotted cotton leafhopper. So they are voracious, and they don't seem to they seem to eat anything. With that, I'm done.
Thank you very much, Lance. Very informative. We have some time for questions, and soon we're gonna do the polls, and I'm gonna also share a link for an anonymous survey where we would like your feedback, and we want to assess what is the status and the situation with Thrips parvispinus. So if you have a question that you want to ask to any of our speakers, feel free to unmute your microphone and ask the question, and we're gonna go through also the questions in the chat. One thing before we move on, again, we would like to apologize for this unforeseen incident, but we appreciate that you have stayed with us today and you followed all the research that has been done and the effort by all the speakers. We are gonna share this recording, the second recording that does not have any malicious activity. We are gonna share it in the Thrips parvispinus webpage in case you missed something. I see one question regarding Metarhizium anisoclea, which is now Metarhizium bruneum, Lalgard, whether we were able to confirm pretty much sporulation on the thrips. And in some cases, yes, we saw that the insects were actually infected by the entomopathogen. This was more prominent in the case of wettable powders. This is where we really saw infection of the insect by the entomopathogen. With the liquid formulations, as I mentioned, we also have this additional mechanical effect on the insect. The oil close the spiracles and then the insects are suffocated, in addition to the entomopathogen that the formulation is already containing. We have a comment about the UV degradation of spores. That is an issue, and that is this is true. And the labels of these products are very detailed. They provide guidance on how they should be applied. The labels state also that you should be performing multiple applications. In our experiments, we did only once, but the labels actually indicate that you need to do a series of applications, and they also provide guidance on the timing of the application and how you should be preparing your solution. There is one question regarding the soil temperature and the nematodes trial during the lab essays. Because we used incubators, the soil temperature was 27 degrees Fahrenheit, so 80 Celsius, so 80 Fahrenheit. This was constant throughout the experimental period. In the greenhouse, the average temperature was around 80 Fahrenheit. There was some fluctuation because during the night the temperature goes down, but the average was 27. This is also the average temperature that we have here in South Florida throughout the year. Of course, there are fluctuations with maximums and minimums, but the average annual temperature for South Florida is 80 Fahrenheit. And this is also the optimum temperature for development and reproduction of Thrips parvispinus. All the experiments that we do are being done in this temperature because this is where the thrips reach its maximum ability to reproduce. For control of short spine thrips in the ornamental landscape, is there a recommendation plan? Currently, like in the same way that we are moving with the nursery production, no. However, we do have, as Livia showed, we do have natural enemies present in South Florida. And then these enemies can be actually, if we enhance their performance, if we try to conserve them, they can help with the mitigation of the pest in the landscape. When it comes down to chemical control, we always recommend softer chemistry, horticultural oils, if it is not too warm, so we don't have phytotoxicity issues, and soaps. From the published chemistry, there are some products that they are registered for use in the landscape, so people can also consider those. But primarily, yeah, you would do biological control with preserving the natural enemies, pruning if you have a heavy infestation. Homeowners can also purchase yellow sticky cards and blue sticky cards from Home Depot or Lowe's, and then they can simply place them in the yards and have different thrip species, including parvispinus, to be caught on these sticky cards. I don't know if Lance, if you have some additional suggestions for landscape management options. You are muted.
Sticking with the horticultural oils, the softer chemistries, they're using a lot of oil and they're not having any problems. Some of the major companies said they're not seeing any phytotoxicity, but you need to try a little bit on the plants you're treating with initially to see if it's safe. That's pretty much it. You can't, unfortunately, the what is it, pylons not legal outside of greenhouses or for use in the landscape. So two or three of our best tools are not legal.
Yeah, we urge the people to follow the label recommendation, because the label is the law. You should always follow what the label says. We have another question. Of the beneficial predators mentioned, are any of them suited for South Florida climate? I would say all of them that were tested, including the Franklin Thrips. All this work has been done in South Florida up to the Orange County, and we have seen them not having an issue with the climate of Florida. I don't know if Lance, you have an addition.
I haven't seen any issues. I mean, we did have the failure of Swirsky during the fall, but that was a field trial on peppers.
There is another question. Can any of the bios be combined in a single application, such as Oreos with Swirsky, or will that result in cannibalism? Cannibalism is when the same species eat each other. What we are not certain just yet. That would be the correct answer. That will be the most correct answer. We do not know if combination of the predators is going to actually give you better results, and if there is compatibility of the predators, just as yet. I don't know if Paola and Livia have some additional remarks.
Yeah, no, I have actually something to add about a comment of Steve Arthurs, that he was surprised that the roof beetle didn't seem to work for Thrips parvispinus. And we were actually also surprised because we know that they feed on soil organisms, but it could be that the temperature would play an important role here. And when I tested it in an incubator. So I placed the pots. It's also related to another question here that was asking about the temperature in the soil. So I didn't measure the temperature in the soil, but we placed the vials in an incubator at 27 degrees. So the temperature in the soil should be warm, which would be realistic to the conditions in Florida, like Alex just mentioned, because we wanted to mimic what the natural enemy would experience in the field outside or in the greenhouse, right? Here in Florida. And it didn't really work. That's why I continued doing replicates to see if we would maybe see something. But it was really clear that the difference between the treatment and the control was not different. So that's why we decided to not continue doing research with the roof beetle.
Great.
Yeah.
So I have
Yeah, it was
this
Carnia.
is a question for you.
Yeah.
They would like
Lace-wing
to know.
species was Carnia. Yeah.
Yes. Perfect. I have included a link for the survey. It's an anonymous survey just for you guys to give us some feedback. Where do you stand with the Thrips parvispinus management? I'm gonna load the polls to do the test. I hope right now everybody can see. After all these talks, let's see, where do you stand with knowledge on this pest? And while you are doing that, I would also like to take the opportunity to thank today's speakers that put a lot of effort in preparing their presentations and giving the presentations. But I would also like to say a special thank you also to the lab members of the Ornamental Entomology Lab here at TREC, because they have been working tirelessly to get these experiments done and disseminate the information to you, but also they assisted today with the organization of this webinar. They have always been there supporting the mission of the lab. So a special thank you to all the lab members as well. So there are 17 questions. We are going to go through them one by one and give you the right answer. And I see that people are reaching now a consensus about their answers. We will email you the CEUs if you have indicated that you needed one. And so you can have it for your records. It might take a few days, so please bear with us until we make sure that you participated in the webinar. And we will also try to make the link, the recording of this webinar accessible very soon. See, already several people have answered. Let's wait a couple of more minutes for everybody to share their answers.
Is there a way to resend it to someone, Alex? Because Michael said that he submitted it before finishing. So he would like to redo it.
Right now, it doesn't give me the option to do that. I leave it 20 more seconds, and then I'm gonna end the poll and share the results. Okay. Let's share the results. So the first question, Thrips parvispinus can feed only on peppers, gardenia, and mandevilla. The correct answer is false. We saw that they can feed on hibiscus and it has a lot of other hosts. Second, Thrips parvispinus is not affected by high temperatures. This is false. The higher the temperature, the faster it's gonna complete the development, but it's gonna have also a reduced reproduction. Number 3, Thrips parvispinus pupates in the soil. This is true. It does pupate in the soil. 4, the second instar larva is the most damaging Thrips stage. It is false, although it might not have been super clear from the images. The first instar larva is more damaging than the second instar. We hypothesize the reason for that is because the first instar really needs to consume a lot of food in order to mature and mold to the second instar. It has larger energy requirements, so it damages the plant more than the second instar larva within 24 hours. Then, a rotation of only spinosad, sulfoxaflor, spinetoram, and chlorphenapir will eliminate the Thrips population. The answer is false. We saw today throughout that integrating different management practices for this pest will yield the best result, and you will be able to achieve a longer-term solution. The best management options to manage Thrips parvispinus are all of the above. So you need to do IPM. You need to implement different strategies, not only one, if you want to be successful. Predatory mites can be used to manage adult thrips. For the predatory mites, no, we don't really recommend them for the management of adult thrips. They target and they prefer, especially the mites of the family Phytozoidae, they prefer first instar larvae. Neoseiulus californicus is the best predator for Thrips parvispinus. This is a predatory mite, and the answer is false. Aureus, which is a predatory insect, can prey on Thrips larvae. This is true. We saw that it's a very voracious predator. Metarhizium bruneum can be used against Thrips parvispinus. The answer is true. We saw that this entomopathogen, and when it is formulated as Lalgard, it can be very efficacious against Thrips parvispinus. Isaria fumosorea can be used against Thrips parvispinus. That is also true. We also saw in today's presentations formulations of this entomopathogen are efficacious against thrips. The entomopathogenic nematodes cannot target Thrips parvispinus pupae because they are too small. This is false. We saw from today's presentations that we can use entomopathogenic nematodes when they are applied in the soil to target the soil living stages. Steninerma felti has the most potential against Thrips parvispinus. Based on our research and the data that we have collected thus far, the answer is false. Steninerma carpocapsa has a higher potential against Thrips parvispinus, and we saw this in the laboratory as well as in greenhouse experiments. Which predator is not legal to use in Florida? The correct answer is Frankliniothrips vespiformis, and also Zwirsky can be used without an issue. What you need to know is that Transeus montorensis is illegal to use in Florida. Which are the ants that can reduce the thrips and mite populations in greenhouses? The correct answer is ghost ants. Dr. Osborne showed you that ghost ants have the potential to prey on thrips as well as mites in greenhouses. Bifenthrin can be used to kill most of the natural enemies of thrips. The answer is true. Bifenthrin is an active ingredient that is not compatible with biological control agents. Which of the following are not known to reduce thrips populations in the greenhouses? The correct answer is bacteria. And bacteria are not known to cause mortality against thrips. And with that, I will stop sharing the results. I'm gonna insert again the link to the survey in case you didn't receive it. We are gonna reach out to you with the CEUs. And once again, we would like to thank you for your patience. Thank you for joining us today. We hope you learned a lot. A big thank you to all the speakers of today and the people that helped organizing this webinar. We hope to see you again in the near future. Thank you.
Thanks.
Thank you, Alex. Thank you, everyone. Have a nice day. Bye.
Bye-bye.